talk about two things. One is the genome of the novel coronavirus, which causes the disease COVID-19, and two, the test for the coronavirus. COVID-19 is actually coronavirus disease 2019. All right, so the test for the coronavirus, I'm going to mainly focus on the nucleic acid-based test, which is RNA or DNA, and the techniques that's used for this test is called PCR. Okay, so how do you find the genome of the coronavirus? What I did was I took this paper published from the University of Texas at Austin in February of 2020, and for their research they used the sequence that was published by the Chinese government, and they have a GenBank ID for the coronavirus genome sequence. So usually any organism when sequence, their sequence is publicly available on NCBI website, and each of those sequences are given a unique ID, and this is ID for the coronavirus. And then you can go to the website here of NCBI, and this is owned by NIH. Here you can paste your sequence and look for the genome of coronavirus. And here, as you can see, it brought me to this site, and here is SARS Cov2 isolate Wuhan HU1 complete genome. And if you want to look at that genome, just click on this FASTA link here. And then, as you can see, this is the actual sequence of the DNA or positive strand RNA. And you can see this just a bunch of letters ATGC. And the sequence is actually 30 kb or 30,000 base pair long. So this is not of much information. So to just show you what kind of genes are coded by the genome, here on this website, this is a very nice illustration of the structure of the genome. So consider this from 5' end to 3' end, the genomic RNA of coronavirus, including all 30,000 base pairs. And these are the different genes that it codes for. You might have heard of the spike protein that's coded from the sequence here to here. It's represented by S. And then there's a nucleocapsid gene represented by N. So once you have the genome sequence, you need to design the primers. Now, the problem is that there are different kinds of coronaviruses. And as you might know, there was a SARS coronavirus in 2003. If you want to design a primer for this test that is specific to current coronavirus, you need to differentiate between the sequence of the previously known virus and the current virus. So this is also a good illustration of the difference here indicated in the red. So right near the nucleocapsid gene, there's a sequence which is different in the novel coronavirus from the SARS 2003 virus. So now that we know where we should design the primer, we can go ahead and design. But CDC, if you go to this website, Center for Disease Control, already has 3 different primers for testing for coronavirus. There's N1, N2, and N3. And these are the region, as we talked about, coming from the nucleocapsid region from the genome right here. Okay, so now that you have the sequence, what you do is you run the PCR. Now also, I forgot to mention that there's another primer here called RNase P. And this is specific to human genome, and this will be used as a control. Now, FDA has actually issued guidelines on how to use these primers and how to interpret the results. So for that, we have, so I'm just switching the screens here. So here is, let me go on top of this PDF. So here is the guideline by FDA. And I'm just going to show you some of the things. So as we discussed, these N genes are coming from SARS-CoV-2, and then N3 is actually universal detection of SARS-like coronavirus. So it's not specific to the novel coronavirus. N3 primer binds to any coronavirus. So if you see an amplification from this primer, it's not necessarily it is from the novel coronavirus, it is from just in general coronavirus. And then here is human RNA-seq information. All right, so you scroll down and you'll see this table here. And this table is just showing what are the expected results of the controls that you need to run along with the test. So negative control here is a non-template control. So you do not have any template. That means you do not have any clinical sample in this particular control. And all you're doing is using the test kit itself, and it should show you negative for all these three genes, N1, N2, and N3, and also for human sample. If you see positive in any of these cases, that means your kit itself is contaminated and you should use a new kit. Now, the kit that is approved by FDA also comes with a positive control. This positive control has basically the DNA from the coronavirus. So when you run this in your test, you should see positive for N1, N2, and N3, but you should find it negative in the RNA-seq primer reaction, because this positive control only has DNA from virus, not from humans. You also are required to have a negative extraction control in negative clinical samples. So this one is from a person who does not have a coronavirus. So you should find no amplification from N1, N2, and N3, but you should see an amplification from RNA-seq because this is the gene from human genome. So these are some of the controls that FDA requires you to have when you are doing testing. All right, so let's look at what the results look like. So in table 2 of this PDF, it shows the interpretation of patient result using this particular kit provided by or approved by FDA. So on top here, you can see this N1 gene, N2, N3, and RNA-seq. What should be your interpretation, report result, and what is the action you should take. So on top here, say you find this particular sample positive for N1, N2, N3, and RNA-seq positive or negative, the interpretation is that you have detected SARS-CoV-2, and the report result should say it's positive, and the action should be report to the sender and appropriate public health authority. Second case, if you have one or both of these N1 or N2, but you either have or do not have N3 and RNA-seq, this also is positive because N1 and N2 are specific to SARS-CoV-2. Sometimes you can have, so the reason you are testing so many different genes is to reduce the possibility of false positive and increase your confidence on the test. So if you have a test result where N1 and N2 are negative, but N3 is positive, which is amplifying any coronavirus, not specifically the novel coronavirus, then it should be interpreted as presumed positive. While in this case here, you don't find N1, N2, and N3, any of those, but it's positive for this human-specific gene, the result should be negative. And then since this is a control, the RNA is P, and if you find this negative along with everything, that means this test is invalid. That means even though you had the sample, clinical sample, which was from a human and you were not amplified, this positive control, this should be considered as an invalid test. So those are some of the things on the genome of the coronavirus, the primers that needs to be designed, or the ones that have been designed by CDC, and the interpretation guidelines for FDA. Hope it was informative. Thank you.